Welcome everybody. You have clicked in a series of teaching slide presentations explaining clinical drug development in oncology. This time we will talk about the modified BOIN design, Bayesian Optimal Interval design for phase 1 studies in oncology. I've been asked by a colleague of mine to make this video. Hi Robert. I will explain it as I would if there was a junior colleague coming to my office for advice when he was new to the industry and thrown into a situation to chair a dose escalation meeting, and the protocol he took over was written in BOIN design. My name is Johannes Wolf. This is recorded in November 2024. This is a classical study design, phase 1A and phase 1B. It has a dose escalation component starting with 25 milligrams and going up to a planned maximum of 350 milligrams, followed by a dose expansion with 2 doses. And this study escalation piece was done with 3+3 design. BOIN design is a substitute for the 3+3 design, and it only affects the dose escalation component. Why would anybody change from 3+3 to something else? 3+3 is well established. There is another video where I explain 3+3, and it should be very clear to any medical director. But it has its weaknesses. Among the weaknesses was that the number of patients enrolled in the lower cohorts that are not effective is too high, and the number of patients in the final cohort that is finally taken as maximal tolerated dose is too small, so that relatively little is known about the final dose. There was a further development of a very intelligent Bayesian design. It was called continuous reassessment model. And it has a model of toxicity in the background that changes with new data entry. The downside of that model was that it is very unpopular because of its complexity, its statistical complexity, a lack of transparency, at least for medical doctors, and the difficulty of medical supervision thus. Out of that were then the model assistant systems. The first of those was the mTPI design, and there is another video about that. And today we're going to talk about the BOIN design. Those model systems are model assisted. The model is there in the beginning, but then it doesn't change during the trial. They are more flexible than the 3+3 design, allow different numbers of patients enrolled in the different cohorts. They're also more flexible during running the trials. They're more powerful than 3+3, but maybe not quite as powerful as CRM, but they still require additional rules. You cannot just say we use BOIN design. You need to describe further things in the protocol. We'll come to this in a moment. Before we go to the details of BOIN design, let me just give you the basic assumption of all of this. Drug development, finding the right dose is a very complex thing. In order to make that mathematically accessible, it needed to be simplified. And there are a number of simplifications you need to keep in mind when you follow those things so that you don't overestimate the final result of these wonderful study designs. There is the single cancer assumption. When doing these trials with those study designs, you kind of ignore that there are different cancer types, different grades of cancer. The cancer has spread in various ways, and that there are different molecular profiles. There is the homogeneous patient population assumption. With that, you ignore that there are patient characteristics that might predict drug toxicity. Now, we're not ignoring that cancers are different from each other, nor do we ignore that patients are different from each other in these study designs. But what we're ignoring is that there are characteristics in the patients that could predict the toxicity. We kind of think that the differences of patients is just random. We have a dose-limiting toxicity, yes or no. And in reality, we are simplifying the grades and types of toxicity to a simple binary measure. We also ignore the relevance of exposure time. And that finally altogether comes to the assumption that there is such a thing as a single optimal dose for all patients. There is no such thing. But without that assumption, you would never be able to build any of those designs. So the key is use the assumptions, find the design, keep in mind that there are assumptions which all of them cannot be true. And then once you have the MTD, go back and become more sophisticated and accurate after declaring the MTD. Those study designs have certain abbreviations you need to know. DLT stands for dose limiting toxicity, and it is bound to explain what type of toxicity is a DLT, and is also linked to a DLT period, which is the time in which DLTs are counted. If a toxicity of that type occurs after the period, it's not counted as a DLT. MTD stands for maximal tolerable dose, and that is sort of the end point of the dose escalation. And it also leads then to a recommended phase 2 dose, which may or may not be the same as the MTD. This is the typical flow diagram of the BOIN design. And it kind of follows in style the classical 3+3 design. Let's do that one just as repetition. You enter 3 patients in a cohort. They all are treated with the same dose. And if none of those experiences a dose-limiting toxicity, then you assume that your dose is too low and you escalate to the next higher dose. If, however, of the 3 patients, there is one that experiences a dose-limiting toxicity, then you think the dose may be just right and you enter 3 more patients with the same dose. If there are more than 1 patient experiencing dose-limiting toxicity, then you assume the dose is too high and you stop enrolling on that cohort, either declare the maximal tolerated dose or go 1 low dose level lower. Very simple. Now it's a bit more complicated, and watch the 3+3 video to see that, but today we go through BOIN. It is very similar. You start treating 3 patients with the same dose, and we call that a cohort. Now, particular to BOIN is that it doesn't need to be 3. It could be a different number. You can determine that in the protocol. Then you determine for each of those cohorts, has the stopping rule been met? And if so, then you determine the MTD. The MTD is not necessarily the last dose that was given. Now, we assume the stopping rule hasn't been met yet, so we actually do our calculations. We calculate the dose-limiting toxicity rate, and that is the total number of patients experiencing a DLT divided by the total number of patients evaluable. Evaluable would be 3 in this case. If that DLT rate turns out to be lower or equal to the number written on the left of the diamond, then you assume the dose is too low and you escalate higher. If it is higher than the number that is written on the right of the diamond, then you assume the dose is too high and you de-escalate. And if it is in between, you continue enrolling on the same dose. Again, different from 3+3, the number of patients to be enrolled is flexible here. The protocol can define it, and it can also be left open. After those patients have been enrolled, you go back and the process repeats. This is the original diagram that you find when you follow the process how to make a BOIN design. You'll see it in a few slides, except for those arrows. Those arrows are my contribution, because otherwise you could go in circles here. Almost all protocols have this BOIN table. When you see that table, you know the study is a BOIN design study. The table gives you what you need to do for any number of patients in a cohort, and a cohort is defined as all patients treated with the same dose. The BOIN design is different from the 3+3 design in the way that it doesn't go up and down and tell you 3 patients to be enrolled and when the entire study stops. The BOIN design asks for the total number of patients you want to enroll in the study altogether. And then this table gives you where you have to go every time you make an assessment. And in that assessment, it could be that the last 3 patients were enrolled in the last round, but before there was a different dose and before there was the same dose. If that is the case, all the patients with the same dose are counted together to come up to this number of the cohort size. Let's assume we have 3. If there is no DLT among the 3 patients, then you escalate. You go to the next higher dose. That's exactly the same as in the 3+3 design. If there is 1 DLT, however, you de-escalate. And that is different from the 3+3 design. This table is not the same table for all protocols. The way this table comes up depends on which variables are entered for the target toxicity probability, and in this case, it's 0.25, 25%, and what the overdose control is. I'll explain those things in a moment. And it's typically described somewhere underneath the table with a little asterisk, very small. It is irrelevant by the time you reach this. By the time you reach this, you have the table you need to follow. Sometimes you have that table with only 2 lines, escalate and de-escalate, and the stay line is left out, but that's very simple. It's just all the numbers that are higher than this one and below this. It's more of a problem when the eliminate one is missing. Let's hope you don't run into this before you've finished with watching this video, because then you can do it yourself. This is the original publication by 2 authors, Liu and Yang. They are both from MD Anderson Cancer Center, and this was written in 2014, published in 2015. And all the coming slides, most of the coming slides are screenshots. Is a study design similar to 3+3. You enroll patient in a cohort, you review the toxicity, and you determine if the next cohort should be higher, lower, or the same by comparing the observed frequency of toxicity with the desired rate. Eventually, when the study is finished, you determine the maximal tolerated dose. Different from the 3+3 design, there are a number of variables to be determined in the beginning, starting with the target toxicity probability. The target toxicity probability is called phi. Now, with that, there is an upper and a lower limit of what is considered acceptable, where they want to stay at the same dose. And those limits are called phi 1, if it's below that, the dose is too high, or phi 2, if it's above that, the dose is too high. So in the beginning of writing a BOIN design, you need to specify phi 1 and phi 2, and the authors recommend 0.6 times phi for phi 1 and 1.4 times phi for phi 2. So in an example that they have of 25%, here we are again, that would end at 15% at the lower limit and 35% as the upper limit. It's a Bayesian method. So Bayesians always go from a prior assumption or knowledge of a probability, then they add the new information and they come to a posterior. Here they recommend a non-informative prior. That means all 3 of them are 1/3. This study design is different from the 3+3 design also in the way that you determine a total number of patients in the beginning, and then you run the design going up and down until your number is reached. And then the maximal tolerated dose is determined from all the data. This determination of the maximal tolerated dose is a process by itself. It is not the same as the last dose that occurred in the protocol. There is something that they call overdose protection, probably because the clinicians told them, this is all too risky. I want to be sure that I'm not treating too many patients with the toxic dose. Oh, the paper carries on saying there is another design. They call that the global design, and they compare the two, but the global design was inferior. And the way they determined that was in part by simulation. Simulation means create virtual patient populations where the computer knows what the MTD is, and then they throw randomly patients out of that patient population into the design and determine how long it takes the design to find the MTD or how close the design comes. And then they do that 10,000 times, and then they compare how good the different designs were, which one was better. And all together, the BOIN design was very good. It was better than the mTPI, not quite as good as CRM, but it is much simpler. So in the conclusion, they call that a flexible framework for conducting interval designs. The paper goes in more details, and there's much too complex to describe it in a video. You feel free to read it. I actually recommend it. It's a good paper to read if you enjoy mathematics. There is one more thing beyond the paper. There is a determination letter by our Food and Drug Administration, our tax dollars at work. The officers, director of the FDA of various types, have written a letter to the senior author of the paper in which they determine that this statistical method is fit for purpose. That's an honor that very, very few people or study designs ever get, and it may be one of the reasons why the BOIN design has become so popular and so well believed. Okay, we've gone through the paper. Now let's assume we have to write a protocol. We have to come up with these graphics and tables. This is how it was in 3+3 design. The design told you what to do. There were no choices. There were no decisions to be made. There were 3 parameters that could be modified, and they determined how aggressive the study was. You have to adjust the study to the disease. A disease that's rapidly deadly, where there is no approved drug. That disease allows to go higher with toxicity, sail higher on the wind. It actually demands that. In contrast, a disease that's not deadly, where there is something out there that helps, and you want to do something better than what is there, you have to be much, much more cautious with how much toxicity you allow. And the study design you write needs to adjust to those things. 3+3 design did allow that. The most important was the definition of the dose-limiting toxicities. If this is a very aggressive disease and you think aggressive treatment is appropriate, then you defined high grades of toxicity as DLTs, and the other way around. The DLT period is an important thing. It is something like 21 or 28 days, and it depends in part in the disease, but also in part on the pharmacokinetic and biology of the drug. There was the maximal dose, that is sort of the upper limit of how much you dare doing, and there is the starting dose, and that comes out of animal data and pharmacokinetic data and is a science by itself. Those 4 things you could adjust, and you could write really good study designs even without a statistician. It is like a mixer with 4 buttons to move. You can buy those in Amazon. That's where the photos come from. BOIN design has the same 4, but you actually can't determine them before you know how the statistical part turns out. And for the statistical part, there are 12 further decisions to be made. Number 1, prior data. This is Bayesian Optimal Interval design. Bayesian needs priors. Default is we don't use priors. It is non-informative. That's how you get over that question when the statistician asks you. That's the first question. Next one, target toxicity rate. Use a default of 30%. Don't get offended. I know we physicians think our target toxicity rate is 0. We want no toxicity. But that's not how this works. You have to come up with a number that describes how much you tolerate. For BOIN design and for mTPI and so on, you need an upper and lower limit of toxicity, what you accept as the range of what is acceptable. That's the phi. And the paper recommends a 0.6 and 1.4 times phi for those rates. You need a total study design size, and a phase 1 study may have 20 to 30 patients. So you can go in the discussion for the first draft of your protocol with, let's say, 30 total study size. The overdose protection, a default may be 95%. The number can go down as low as 60%. And the minimal and maximal cohort sizes, you may use defaults of something like 3 to 10 or 3 to 12. With those answers, the statistician can give you the study design. But it's almost certain that when you then get it back and you look at it in detail, you may not like it. So here we have an advantage. We have software that allows us to look at what the effect of our answers will be. So I'm an experienced clinician. I have a good gut feeling for how much toxicity I think is tolerable for a deadly disease. But a target toxicity rate is nothing for which I have a gut feeling, or at least didn't have it when I started. What you can do is put a number in, see what it turns out, then look at the outcome of that, compare that to your gut feeling, and if you don't like it, change the number until you somewhat like it, and that will then be your answer to your statistician. Here's the software. If you Google BOIN and MD Anderson Cancer Center, MDACC, this is what you get at least now in November 2024. The top line, if you click on that, you come to this study designer. I've tested a few of them to show you and raise your appetite of playing with it. So here's the flow diagram that we showed before. Here I've entered the target toxicity probability of 0.1 versus 0.2 versus 0.5, and you see this number and that number changing. If you only allow a target toxicity profile of 10%, then 0.04 is the number below which you think it's too low. Otherwise, it's 0.19. That's the standard example. And if you go higher and allow half of the patients to have dose-limiting toxicities, then 0.39 is the lower limit that is allowed in the decision diamond. In the same software, you also get the table. And again, playing around here, I've done the same examples, and here I have the table. Let's look at one example. So, target toxicity profile of 0.1, 9 patients enrolled. Obviously, if no patient has a dose-limiting toxicity, you escalate. But if there is only one patient with dose-limiting toxicity, you don't escalate, you stay. And with 2, you even de-escalate. Target toxicity profile of 0.25, 25%. That was the standard example before. And those are the same numbers we looked at before, with 9 patients enrolled, 1 patient escalate, 2 patients with DLTs stay, 3 patients with DLTs de-escalate. If there are 5 patients with DLTs, then you eliminate the dose level. And if you enter a target toxicity profile probability of 0.5, you allow half of the patients to have dose-limiting toxicities, then 3 patients will still allow you to escalate, and only 6 patients get you to de-escalate. So that makes sense. In all of those, I've put the overdose control at 0.95, 95%. There is also a graphical design that I actually like a lot better. The problem with this design is that most people, when they look at a table like that, they think it's mTPI design. mTPI design is the older one that led to BOIN design, the one that broke the 3+3 dominance. The same software will give you this table, and let's just look at it. Those are the same examples: the number of patients with DLTs on the y-axis, the number of patients that were enrolled, the denominator on the x-axis. So 9 patients enrolled, same example before. If all 9 have a dose-limiting toxicity, then you get DE, that is de-escalate and eliminate. Never ever use that dose again. Same down to 3 for that target toxicity profile. If you have 2, you stay. No, sorry, you don't stay. You de-escalate. If you have 1, you stay. And if there is none, then you go up. And if you allow 50%, then the elimination goes up to 8. So this all makes sense, and it's a wonderful way to look at it. Now we're keeping the target toxicity profile at 25%, but we're changing the overdose control. This is what we've done before, overdose control of 0.95 on the right as example. And this is the same graphic you've seen there. If you increase the overdose control, then more of the numbers here turn into elimination. Actually, the de-escalation completely disappears. You have 9 patients enrolled, and if 3 or more have dose-limiting toxicities, you never go back to that dose. The dose is eliminated. That is what happens with the overdose control. Now, I think there is a glitch in the software. Never trust software all alone. With the same data entries, this is the diagram I'm getting. And you see, it gives you 0.195 for that target toxicity profile and that overdose control. We've had that many times. If you look at 17 patients enrolled, 5 patients had dose-limiting toxicities, that gets you a DLT rate of 5/17. That's 0.295. That would mean stay. If you look at the table, that's correct, because that number is between that number and that number, so you go down and stay. But you get the same flow diagram when you enter those numbers. And here, 5 patients would have called for de-escalation. I think there's a little glitch, and that just says, be careful, even with software that comes from MD Anderson. Now, the same software, isn't that amazing? That same software gives you template text for writing your protocol. I would give them a Medal of Something honor. These people that make this, they really make our life as easy as they can make it. It's written by the statisticians at MD Anderson Cancer Center. After you put all the numbers in there, you can download that text and you can put that text into your protocol. We're coming to the end of my presentation, but please do not let that presentation be the only way how you learn about BOIN design. I recommend to read the paper, the FDA letter, to watch more videos. If you like watching videos, those 3 were the ones that I liked best when I prepared for this video. This is from one of the authors, and it is from MD Anderson Cancer Center, and it has been reported shortly after the paper was published. This is a teaching video, a real formal class, an online class. This is also a class, but it is a lecture where students ask questions, and this is more structured. And finally, this is from Dana Faber, just to say that MD Anderson Cancer Center is not the only cancer center on the planet. And here is a very good discussion in a team where people ask questions and she answers and tells you what the benefits of BOIN design is and where you actually don't need it. Disclaimers and the conflict of interest. This video was brought to you by Oncology Consulting Wolf LLC. The company is solely responsible for its content. In general, the consulting company works with various pharmaceutical companies with fee for service, but this video has no sales contracts, earns no sales commission, and there are no further sponsors and no external influence on the opinion of the author. Science moves on. The video reflects the opinion of the author in November 2024. Opinions may change and science moves forward. While the author has a medical license, producing or listening to this video does not constitute a physician-patient relationship, Any recommendations that may sound as medical advice are general statements and should not be followed unless discussed with a real medical provider in person. I hope you enjoyed it. Thank you very much. Bye-bye.